In today's video at Medical Sciences, we want to discuss about the structure of the eukaryotic cell, and the function of each part. So we're going to look at different parts of the eukaryotic cell and then their function. That's what we are going to look at in this session. And if you are new and you haven't subscribed, please go to the subscription button and make sure to subscribe so that you're always notified whenever we release other videos. So today we are discussing on the structure of the eukaryotic cell, and this structure, it contains the three basic parts. And before we begin the structure, we know that the cell is the functional unit within the body. So a cell is the functional unit, and if we have a collection of cells, we form a tissue, and the collection of tissue forms an organ, then an organ, the collection of organs form an organism. And the three basic parts, we shall have the cell membrane, we have the cell membrane, we have the cytoplasm, and the nucleus. So if they ask you the three basic parts of the cell, these are the basic parts, whereby the cell membrane is the one that gives the shape to the eukaryotic cell. So it is the one that separates the internal structures of the cell with the external structure. And it is selectively permeable, as we shall see its structure in another video, whereby this cytoplasm is made up of a phospholipid bilayer. The cell membrane is made up of a phospholipid bilayer, only allowing lipid-soluble components to cross. And then it is also made up of proteins. And these proteins, according to fluid mosaic model, some move across and they act as ion channels or carrier proteins, whereas others act as receptors. So these are the different components. Then it also contains cholesterol, as we shall see it in another video. Then the cytoplasm, we are going to see that is divided into two, that is the cytosol and the organelles. And the organelles include all these organelles found within the cell except the nucleus. Cytosol is the fluid free from the cell organelles, is what we call the cytosol. Then we are going to see also the nucleus, whereby in the cell, so this is the cell membrane we have talked about, cell membrane, or you can call it plasma membrane, the one which gives the shape of the cell and prevents, it allows the movement of materials in and out and communicates to the surrounding area. Then the next thing we are going to see on the cell, we're going to see the organelles. Inside the cell membrane, we have the cytosol. We have the cytosol, whereby we have said the cytosol is the plasma membrane. It is the cytoplasm without organelles. And this cytosol contains solutes, which are dissolved in this cytosol. Then we have said organelles, cellular organelles include, we have the peroxisomes, we have lysosomes. These are the organelles in the cytoplasm. We have the ribosomes. Ribosomes, we have those which are free, that is in the cytosol, and those which are found on the rough endoplasmic reticulum. Then we have also the powerhouse known as the mitochondria. This is the mitochondria and this is the mitochondria, as we shall see in details in the coming time. Then we also have the Golgi apparatus, or what we can call the Golgi complex or apparatus, is also there. And among the organelles, we have also the rough endoplasmic reticulum. Then we have also the smooth endoplasmic reticulum, that is without ribosomes, whereas RAF has ribosomes. Then we have also the nucleus, and the nucleus, which is also another part, another basic part. It is the nucleus has the nuclear envelope. It has the nuclear envelope. Has a nuclear envelope. Then within it has the nucleoplasm, and this nucleoplasm is made up of chromatins. Made up of chromatins, and these chromatins are the ones that have genes for heredity. Genes are found there, and then the proteins known as histones are also within the nucleoplasm. It has histones, whereby histones we have what we call the chromatins are divided into, whereby we have the chromatin and the heterochromatins. And this is the chromatins are the one that contain chromosomes, and the chromosomes have genes that are responsible for heredity. Then it has also the nucleolus. It has the nucleolus, whereby this nucleolus we are going to see its function is responsible for synthesis of ribosomal RNA, and also it contains DNA. So that is the synthesis for ribosomal RNA that commands the formation of ribosomes, which are found on the RAF and also on this in the cytosol. So, and we have said that the RAF endoplasmic, we call it RAF because it contains ribosomes. So these are the different structures. Then we end with the cytoskeleton of the cell, cytoskeleton of the cell. And this cytoskeleton contains guys like the microfilaments. These are microfilaments like actin and myosin. Then we have also the microtubules and intermediate filaments. So microtubules, we are also going to see their function. So after seeing the structure of the eukaryotic cell, now we are going to see the function of each one by one. And we are going to begin with the nucleus, whereby the nucleus we have said it is surrounded by the nuclear envelope. And this nuclear envelope has smaller pores. And these pores is the one that allow movement of the new nuclear materials out of the cell. So it has the pores. This nuclear envelope contains smaller pores where the RNAs and the replicated DNA after transcription pass through. So we are going to see these smaller nuclear pores are the one that allow movement of messenger RNA, ribosomal RNA, and after transcription to pass through to go into the cytoplasm. And then the nucleoplasm, we have said it contains chromatin, and this chromatin is the one that contains genes, and genes are the one that are responsible for heredity. And also we have said it contains the DNA. These genes are within the DNA, whereby it can perform what we call DNA replication. There is DNA replication to form more DNAs, and then it also performs what we call transcription. This transcription enables the formation of messenger RNA. Then there is also production of ribosomal RNA in the nucleolus, which are responsible for formation of ribosomes. So the two major important functions, as we normally say that the nucleus is the one that controls all cellular activities. Note that all cellular activities are coordinated by the nucleus, whereby it is commanding the cellular activities in two ways. That is by DNA replication and transcription. Whereby DNA replication here we are making many copies, then we also convert them into messenger RNA, which is responsible for translation after passing through the nuclear pores and going to the ribosomes, where the ribosomes act as the machinery for formation of proteins. So those are the major two functions of the nucleus, whereby we have seen it is DNA replication and transcription, hence it is able to control all cellular activities. Then after looking at the nucleus, this nucleus, the next thing we look at is that the messenger RNA produced and the protein and the protein is produced by the nucleus, they move directly because we find the rough endoplasmic reticulum and the smooth directly continuing with the outer envelope. So this is the rough endoplasmic reticulum, which continues with the membrane of the nucleus, and the products from the nucleus go into the rough endoplasmic reticulum. And this rough endoplasmic reticulum, we said it contains ribosomes, and these ribosomes are the one that act as machinery. That if I have ribosome in the cytosol, for example, these are the ribosomes, they are going to receive the proteins or the RNAs, the messenger RNA, and they carry out translation to form proteins. So the major function of the rough endoplasmic reticulum, as we're going to list it here, is protein synthesis. It is responsible for protein synthesis. It is the site for protein synthesis due to presence of ribosomes, which act as machinery after the nucleus producing messenger RNA. That they receive these messenger RNAs from the nucleus and they use them, whereby these messenger RNAs will be having the codons, the anticodons, which are responsible to command protein synthesis to produce polypeptide chains. So the major function of the rough endoplasmic reticulum is for protein synthesis, whereby it is going to produce proteins, produces proteins either responsible, these proteins either will go to the lysosomes, or some are going to form part of the cell membrane, and others are going to be excreted. So those are the proteins which are formed from the rough endoplasmic reticulum. And after forming these proteins, they bud off from the rough endoplasmic reticulum in the form of a vesicle. So the vesicles which bud off from the rough endoplasmic reticulum, they are going to be received. These are the vesicles which bud off. So these vesicles are received by the Golgi apparatus or the Golgi complex. But before we look at the function of the Golgi complex, we have also smooth endoplasmic reticulum. And this is smooth, for it lacks ribosomes. It lacks ribosomes. So for it contains enzymes responsible for lipid metabolism, whereby it is responsible for synthesis of lipids, lipids like fatty acids, cholesterol, and phospholipids. So that is the smooth endoplasmic reticulum containing enzymes responsible for synthesis of lipids, like cholesterol, whereby cholesterol we can use it for manufacture of steroid hormones and fat-soluble vitamins. Then phospholipids and fatty acids are also synthesized in the smooth endoplasmic reticulum. So note this as a student, that the rough endoplasmic reticulum has ribosomes, and therefore it is a major site for protein synthesis. Then the rough endoplasmic reticulum lacks the ribosomes, and it is responsible for synthesis of fats, that is, fatty acids, phospholipids, cholesterol, which these guys can act as precursors for formation of other compounds like steroid hormones and fat-soluble vitamins. So those are the functions of the smooth endoplasmic reticulum. And apart from synthesis of fats, it also contains enzymes that are responsible for detoxification of drugs. So we're going to see detoxification of drugs can also happen in the smooth endoplasmic reticulum. If I can write it here, smooth endoplasmic reticulum is responsible for lipid synthesis, it is responsible for detoxification of drugs, it is responsible for storage, it acts as a store of calcium ions. So calcium ions are also stored within the smooth endoplasmic reticulum and are excreted from there in case needed in the cytoplasm. But the storage of calcium is the smooth endoplasmic reticulum, and also it is responsible for metabolism of glucose 6-phosphate enzymes, whereby the phosphate group is removed within the smooth endoplasmic reticulum. So lipid synthesis, we said we can synthesize the fatty acids, cholesterol, and other components like phospholipids, which can be part of the cell membrane. And these ones, we can use them as a precursor for synthesis of steroidal and fat-soluble vitamins. So this is the major function of the smooth endoplasmic reticulum. And the products of the smooth endoplasmic reticulum, for example, the fats, are also received by the Golgi apparatus or the Golgi apparatus, depending on how you pronounce it. So rough endoplasmic reticulum protein synthesis, then smooth lipid synthesis, detoxification, storage and excretion of calcium, and then metabolism of glucose 6-phosphate. Note those four importances of the smooth endoplasmic reticulum. And we are saying the products of rough and smooth are received in form of vesicles by the Golgi apparatus. So this Golgi apparatus is the one that receives the products of rough and smooth endoplasmic reticulum and receives it in form of vesicles, and the side that receives, this is called the cis face. It has the cis face, then the exit side is what we call the trans. So this is the entry side, and this is the exit, which we are calling the trans face. So after vesicles battle from the rough and the smooth, they are received inside the Golgi apparatus, and within the Golgi apparatus, we are going to see major modification when the proteins and lipids are accepted or enter into the cis face of the Golgi apparatus. They undergo modification. They undergo modification. They are modified to form glycoproteins, lipoproteins, glycolipids, and after forming glycoproteins, glycolipids, and lipoproteins, they are packaged. The next thing it is, before packaging, they are sorted. The middle part, the intermediate part of the system of the Golgi apparatus is responsible for sorting. After modification to form functional proteins and functional lipids, they are sorted. Those which are not well modified, they are taken back in the cis part to be modified. Then after being sorted, then we package. They are packaged. And after being packaged, they are going to bud off. After packaging, they bud off as also vesicles, whereby they bud off. And these vesicles that bud off from the Golgi apparatus, they are taken either to the lysosomes to be digested, or these are the vesicles. Some are taken to form part of the cell membrane, and some are excreted outside the cell by exocytosis. So some are excreted, some form part of the cell membrane, and others are digested by the lysosomes. These are the lysosomes which can digest the proteins released from the Golgi apparatus. So the major function of the Golgi apparatus is to receive the vesicles from rough endoplasmic and smooth endoplasmic. After receiving, it carries out modification to form lipoproteins, glycolipids, and lipoproteins. Then after forming lipoproteins, glycolipids, and phospholipids, then they are sorted. It does the sorting and packages those ones which are well modified. That is the function. So packaging. So if you are in a factory, the place where we package the processed materials is what we call the Golgi apparatus. So it is the place of packaging. So after looking at the importance of the Golgi apparatus, this one is going to lead us into another part of the cell which we call the mitochondria. And the mitochondria, we have zoomed it here. This is the mitochondria. If it is one, you call it the mitochondrion. And this mitochondria is made up of the outer, it has the outer layer, outer membrane, and it has also the inner layer. And the outer is permeable to many substances, whereas the inner is impermeable. And this inner is folded to form what we call crystal. Forms, that's why we say it is highly folded to form crystal, which increase the surface area for importance. And this inner mitochondrial membrane, along the crystal, is where we see many importances of the cell of the mitochondria happening, whereby we are going to see the electron transport chain occurring along the inner mitochondrial membrane, and even then within the matrix we see also Krebs cycle. But this electron transport chain is the one responsible for oxidative phosphorylation to produce energy in the form of ATP. So energy in the form of ATP is produced by oxidative phosphorylation along the inner mitochondrial membrane by the process which is carried about by the electron transport chain. Then remember also that within this same, there is also enzymes responsible for Krebs cycle. And this Krebs cycle also can produce ATP by substrate level, at substrate level. At substrate level, we also produce ATP, water, and carbon dioxide. So we are going to see that the major function of the mitochondria is production of energy in the form of ATP. So after seeing that the mitochondrial outer membrane and the inner membrane, it also contains the mitochondrial matrix and the mitochondrial DNA. It also has mitochondrial DNA, and this is from the mother. The mitochondrial DNA is acquired from the mother, and it is also that controls activities within the mitochondria. But remember, the brain and red cells lack the mitochondria. And in the cells where there is much energy needed, there is increased number of the mitochondria. So within the mitochondrial matrix, we are seeing Krebs cycle occurring to produce ATP at substrate level. Then along the inner mitochondrial membrane, the crystal, we see oxidative phosphorylation by electron transport chain to produce also energy in form. So this is the generator or the power provider of the cell. So energy to perform all cellular activities is derived from the mitochondria. And after looking at the function of the mitochondria and its structure, now we can also go to another type of cellular organelle, and this is the lysosome. This is the lysosome. Lysosome in the cell, these are the ones, this is the lysosome, this is the lysosome, and this is the lysosome. And these lysosomes contain enzymes such as proteases, it contains nucleases. This same lysosome contains lipases and oxidases. So it contains all types of enzymes, and these enzymes are responsible for digestion or breakdown. They break down macromolecules, whereby you have macromolecules like proteins are broken down by proteases. These ones are broken down by nucleases, lipases, and all these they act by hydrolysis. Then, apart from breakdown of macromolecules, these lysosomes are also responsible for breaking down worn-out cell organelles of the cell. They are destroyed or broken down by the lysosomes, and this is what we call autophagy. Auto means self. Then this one is from the word phagocytosis. So it is going to form a phagosome. Then if there is any cellular organelle which is old or which is worn out, it forms a phagosome and then kills or destroys this organelle by autophagy. Then if the cell also matures and is no longer needed, it can also destroy it, and this is what we call, it can also perform what we call autolysis. That is killing the cell where it is found. So this one is, autolysis is breakdown of the cell, the whole cell, where even the lysosome is found inside it. That is what we call autolysis. So these are the functions of the lysosome enzymes found in the cytoplasm. Then another guy found in the cytoplasm is known as the peroxisome. Which looks like lysosome, but this one contains major enzymes, and the enzymes which it contains majorly include catalase, it contains oxidases and catalases. And these enzymes are the ones that are responsible for prevention of free radicals. They prevent accumulation of free radicals or oxygen reactive species. These free radicals once produced free radicals like hydrogen peroxide, superoxides, hypochlorous, whereby catalase is going to catalyze the hydrolysis of hydrogen peroxide to form water plus oxygen, which are not in the presence of peroxidase or catalase. So this one is not toxic, but whereas the hydrogen peroxide is very toxic. So these enzymes enable us to prevent accumulation of these oxygen reactive species like superoxides, the hydrogen peroxide, and hypochlorous, which can destroy the cells. So that is the function of the peroxisome. Then the ribosome, the next is the ribosome. This is, we can name it ribosome. And we saw ribosomes, they are those that are found on the rough endoplasmic reticulum, so those are membrane bound, and we have those ones which are free, found in the cytosol. And the major function, this ribosome is contained, it has what we call large subunit and small subunit. And the large subunit, so when the nucleus produces messenger RNAs, they come and bind into the site, into the ribosome, which acts as the machinery, whereby we have the exit site, the P site, and the A site. In the eukaryotic cells, this one is 60S, this one is 40S. Then in the prokaryotes, 50S and 30S ribosomal subunit. And the major importance of these ribosomes is the translation, protein synthesis, what by the process known as translation to form polypeptide chain. That is the function of the ribosomes in the cell. Hope you people you're getting these concepts. Then the last one is the cytoskeleton of the cell, whereby the cytoskeleton of the cell contains what we call microfilaments. We have the microfilaments which contains actin and myosin. And actin and myosin, this is responsible for contractions. Responsible for phagocytosis and even diapedesis, movement of cells. So diapedesis, muscle contractions or cell contractions, and phagocytosis are done by the actin and myosin, whereby the actin during muscle filament theory, they keep contracting and relaxing. That is the function of the microfilaments. Then we have also the intermediate filaments, and these intermediate filaments, for them they anchor cells. They are the one that can anchor two cells together and also enables the support of the cell together with the outside environment. So this one acts as anchor or support, offering support. Then the last one is what we call the microtubules. And microtubules play a big role during cell division, whereby during cell division, these microtubules are the one that attach the chromatins or the chromatids at the equator of spindle. So if I have a cell, for example, I have a cell here, and we find these are the asters, this is the end poles, we find that these microtubules are the one that are attaching the sister the chromosomes on the equator of spindle. So they attach the chromosomes at the equator of spindle, and they are the one that pull or separate the sister chromatids towards the opposite poles. That is during cell division. That is what you can pick on this cytoskeleton. So this is what we can discuss in this session about different structures and their functions, whereby we have looked at each part and the function it plays in the body. Hope you have gained a lot from this session. Thank you so much for learning at Medical Sciences by Naftali Muhumuza. May God bless you.